Hello, and welcome to session 3 of our discussion of bone tissue. As you know, we're dealing with chapter 6, which is all about bone tissue. And I think we're going to finish up the lecture series with this presentation here. As you recall, as we were talking in last session, we were discussing two different ways in which bone is produced. One of those ways we had talked about, which is being shown here, intramembranous ossification. And we'd gone through our notes and described step by step the process. So let's just quickly review that process using the images from the textbook. And then we're going to go and talk about the second process, which is endochondral ossification. So here, as you can see, step 1, the development of the ossification center. Osteoblasts secrete organic extracellular matrix. And then the next step is we're going to get calcification. Calcium and other mineral salts are deposited, and extracellular matrix calcifies, hardens. So we're beginning to form now bone material. And the osteocyte is now trapped within that matrix. And then mesenchymal cells will condense on the outside of the forming trabeculae, and we have the formation trabeculae. Extracellular matrix develops in trabeculae, and then fuse to form spongy bone, and then development of the periosteum and mesenchyme at the periphery of the bone develops into the periosteum or endosteum, depending on where we are in the bone. So this is a condensed summary of all the notes that we put down in our textbook. So I just wanted to remind everybody. So the deal with this is that we are going to build our bone tissue on a model of fibrous connective tissue. And so many of the bones, in fact all of the bones in the skull and several bones outside of the skull, are all built in this fashion. The other possibility, if we want to build bone tissue, is to go through what is called endochondral ossification. And so that's what we're going to look at now. And again, what we're going to do is step by step. We're going to take notes and identify the major processes that your body goes through to build bones in this way. So this is endochondral ossification. Endochondral ossification. The key characteristic here is that there is the replacement of cartilage of cartilage model, I should say, not just cartilage model with bone. So in the previous process, we had talked about the replacement of fibrous connective tissue with bone. Here we're talking about the replacement of a cartilage model and replacing it with bone tissue. So let's go through the steps. Number 1, we're going to develop. So the development of the cartilage model. Obviously, if we're going to replace this, we have to build it first. Okay. All right. And there's a number of processes that have to go here. So at the site of bone development, specific chemicals signal the attraction or the condensation, let's put it that way. Mesenchymal cells. So they'll condense in a specific area due to the chemical environment. So the more we find out about the human body, the more we see that a lot of what goes on inside the human body is determined by specific chemicals that are being released by cells and tissues, and they influence the development, the process, the growth, and many other aspects of your biology and physiology. So at the site of where we're going to build a bone, there are chemicals that are being released that cause mesenchymal cells to condense within that region, okay? And they will condense into the general shape of the future bone. Remember, this is happening around the 4th week of development, so you're not very big. But nevertheless, you can see areas in the body at this point where bones are beginning to take form, and you can begin to see the outlines of bones that will later be able to easily be identified, such as the femur and the tibia and so forth and so on. Okay. The mesenchyme cells become chondroblasts. So, once again, this is all being orchestrated by a chemical mixture that's being released by cells, and it's a complicated dance. But as these chemicals are being released, changes the behavior of the mesenchymal cells. They start differentiating, and they become chondroblasts. The chondroblasts then start to produce a cartilage matrix. So this is going to be hyaline cartilage. That's the type of cartilage we're talking about. As this process proceeds, the matrix surrounds some of the chondroblasts and they become chondrocytes. So this should be a kind of familiar process. We talked about this in the last process with intramembranous and how the matrix that's being laid down begins to trap the osteoblasts. They become osteocytes. The same kind of thing is happening here, except that in this case we're talking about chondroblasts, and we're looking at the development of a matrix of cartilage that surrounds these cells. But it's the same process. Okay, matrix surrounds some of the chondroblasts, and they become chondrocytes. Around the outside of the cartilage model a membrane forms. Okay. And this we will call the perichondrium. So we know about the periosteum, peri meaning around, osteum meaning bone, And in this case, what we're doing is surrounding cartilage, so it's the perichondrium. Okay. Again, this is all being orchestrated by a chemical environment. This cellular fibrous membrane is going to provide cells for the production of new cartilage and growth. So what we've done is we've started the process of building the model, and we're surrounding the cartilage with a layer, a perichondrium, and that perichondrium then will be the source of new chondroblasts, which will lay down more matrix. They eventually become trapped, but the membrane that surrounds this model is a flexible and a growing membrane, so it keeps up with the growth of the model, and it simply continues to add more and more cells. So it's sort of a source for the stem cells. It is the location of the stem cells for chondroblasts. Okay. All right. Let's see. The second step, then, is growth of the cartilage model. So once we've kind of start just the basic foundation of the bone and cartilage, we're going to grow that. And we have two ways in which it grows. So we have growth in length, and that is interstitial growth. This is in length. And number two, we have growth in thickness, and that's going to be appositional growth. And so that is in thickness. So if we were to draw a little model at this point of our bone, looking something like this, we're going to grow in length, and we're going to grow in thickness. And the interstitial growth is growth in length. Okay. And appositional growth is growth in thickness. Okay. So two different, although related, processes are going to increase the size of the model that we're building. So, growth in length. And again, this is the interstitial growth. Cell division of chondrocytes. And the production of more cartilage. matrix. So that growth in length is going to be the result of division of chondrocytes and also the production of more cartilage. So the chondrocytes, as you probably are thinking, well, those are trapped inside the matrix, but there are some that are still capable of cell division. And remember that the matrix here is not calcified minerals. It's more pliable than that. It's more like a gel, a semi-gel, or a liquid gel-like material at this point. And so cell division is possible, and when that cell division occurs, you're adding new cells. So you're kind of stacking the cells. So if you start off with 2 and you have cell division, then you have 4, okay? And that's a growth in length. That's what we're talking about when we talk about cell division of chondrocytes. Production of cartilage is pretty clear. We're simply adding more of the matrix to grow in length. Okay. Growth in thickness. And once again, what we're talking about is appositional growth. Okay. And what we're going to do is we're going to grow through the addition of new matrix on the surface of the model, okay, from new chondrocytes originating from the perichondrium. So if we were to draw, and this is our matrix, and we can come in here, and I'm drawing it with some space here, just so I can get some, we're going to add new cells here. So new cells are originating from this, and we're adding more material as that process goes on. This layer, remember, is a cellular layer. It's not like a piece of saran wrap or plastic wrap that is surrounding the bone. It is a living membrane, and it's capable of adding new cells to increase the number of cells that make up that membrane. And so as the model is growing and moving out, this layer is also adapting to that growth by creating new cells and allowing that layer, the membrane, to grow along with the growth of the model itself. And there's a continuous supply of cells that are going to be adding new matrix as that process happens. And we're just going to keep growing in thickness from this layer. And the layer is adapting. So it's not like we just have a one-size membrane, and so we're just simply stretching that. No, these are living cells. They can divide, they can grow, the membrane can grow and respond to the change in size of the model as it grows too. Okay. All right, so the addition of new matrix on the surface of the model from new chondrocytes originating from the perichondrium. Okay. As the model grows, chondrocytes in the mid region of the model enlarge. And the term that we use to describe that enlargement is hypertrophy. So they're enlarging. And let's, at this point, I think it's worthwhile to look at an image, a picture, to help us visualize what we've just described here. So we're going to do that. And if we can zoom in here. So here we are. This is the point at which we are now. You can see that they have indicated there's perichondrium. They haven't drawn it in very well. I wish they had done a little bit better job of that. But what you're noticing here in the center here is these cells are enlarging there. Okay. Now, keep in mind, we're continuously adding new material to the outside. So these cells are buried, are being more and more buried within that matrix. And since they are buried, it's becoming more and more difficult for them to get nutrients and oxygen and to get rid of waste. And so their response is to grow bigger, that is to enlarge, and push the matrix away from them so that you actually create a larger cavity in which these cells are living. But they're also suffering and they're beginning to die. They will die. And we'll talk about that in just a bit. So when we get to this point, we'll talk about that. So this is development of the cartilage model and then growth of the cartilage model. We haven't yet got to this, which is the periosteum. So we're going to build a bone collar, but that's in our next step. So we'll get there in just a bit. All right. The surrounding, so as that growth is happening, enlargement, hypertrophy happens, the surrounding matrix begins to calcify. It begins to harden. So coming back to the image, you can see calcified matrix, uncalcified matrix. So this area where the cells are enlarging, the matrix is hardening now. It's no longer that semi-jelly sort of substance. It's hardening, and that makes it even more difficult for materials to get into those cells so that they can get the nutrients and the oxygen that they need. Okay? So a lack of nutrients due to, number one, the calcifying of the matrix, and number two, a bone collar, which we have not yet described, but that's going to come shortly, and that's also going to increase the difficulty of material getting into the center of the growing bone model. So calcification is the first step, and then later, as we'll see, we're going to grow a bone collar around the center of that bone. That's going to cause cell death, okay, of chondrocytes. The cell death itself is going to add to the whole problem of nutrients and causing the calcification of the matrix. It's going to change the internal environment there. It's going to cause further cell death. So what we're really setting up here is a positive feedback cycle, where as cells die, they cause more cells to die. And it's just going to feed on itself in the center of this model. Okay? The spaces left behind by dead cells merge. So these large openings then begin to merge and form larger cavities. Okay. And then we will start to identify them as lacunae. Okay. Now we've set the stage. We've got a growing model. We have cell death starting to take place in the center of that model. We have hardening and calcification of the matrix within the center of that model. And that cell death is leaving behind large spaces. So the next step is number 3 here, and that would be the development of what is called the primary ossification center. Okay, and I'm going to go ahead and that one doesn't help me. I was going to look at another picture, but I can see that that's not going to help us here. So, development of the primary ossification center. To review, we're at we've started here, and we're approaching this step right here. So here I fault the textbook a little bit because I think they should have had another picture right here between these two. So this would be the beginning, and then showing this without the bone collar, because this is conflating actually two steps in the development together. Yes, this is true. We have the growth of the bone model, but this formation of the bone collar would happen after this process has already begun. And so I would have liked to have seen another picture in between here without the bone collar and just simply what we see here in blue being depicted, and then that picture there. So we're headed towards this picture right here now. That's what we're going to be talking about. Even though they call that the growth of the cartilage model, what we're now going to describe in step number 3 here is what is being pictured at this point right here in the drawings. Okay. So development of the primary ossification center. Cells from the perichondrium differentiate into osteoblasts. So once again, that layer around the model is going to be a source of cells that are going to change the nature of that model. And in this case, we're going to now have osteoblasts. And once again, this is the result of chemical cues that are being released in a step-by-step process to change the developmental pathway of certain cells. Okay? So cells from the perichondrium differentiate into osteoblasts. They begin to form a collar of bone tissue around the center of the shaft of the model. This will be called the bone collar. Okay. So now we have an accurate picture. So now the textbook, what they call the growth of the cartilage model, is now picturing this situation here. And this is where we are right now. So we have this collar of bone around the shaft here. Okay? And as you can imagine, that's going to even make it more difficult for materials to diffuse into the center to feed these cells that are now trapped within that collar of bone. So that it's just adding to their woes and making it more difficult for them to survive. In the next step, what's going to happen is the perichondrium around the bone collar becomes the periosteum. So it's no longer called the perichondrium because it's surrounding bone now rather than cartilage, and it's called the periosteum. Okay. And this is now going to be populated with osteogenetic precursor cells. So the name change is not just because we are now surrounding bone, but the nature of that membrane also changes. And instead of supplying chondroblast, it will now start to supply osteoblast. And so the osteogenetic precursor cells are now part of that membrane, and they will give rise to chondroblasts. Okay. The formation of the bone collar limits the ability of nutrients to diffuse into the model, just as we've been talking about. Okay. This will cause further calcification of the cartilage matrix and more cell death. Of chondrocytes, just to make sure everybody understands what we're talking about, what cells we're referring to. Okay. Now, this process of cell death, calcification, is going to set off now a secondary result, which is that we're going to start recruiting blood vessels to merge into this region. So these processes signal recruitment of a primary blood vessel. Okay. So what is the primary blood vessel in the shaft of a long bone? The nutrient artery. Okay. And this will enter, so the nutrient artery enters into the calcified cartilage. It grows into that space. Okay. It penetrates the bone collar. It penetrates the bone collar. And grows into that space with the help of osteoclasts. Where did the osteoclasts come from, you may ask? They are coming from the blood that is being carried in this artery. So if we were to draw, let's see if I can do this some justice. And let's bring in, this is a magnified view. And what we have here is, we'll create some spaces here. And then this would be the bone collar on the other side. And this is our bone. Our bone collar. So what's happening is, as that blood vessel comes in, it's going to need to go through the bone. How does it penetrate the bone? Well, inside the bone are osteoclasts. Okay, so recall they're being produced in as part of a subset of cells that are in the red bone marrow. So they're being supplied, and these cells come into this region, and now they are going to start what? They're going to start eating their way through not just the bone, but the cartilage itself. That's calcified cartilage. So we're going to start clearing out a space, because we have brought in that blood, and the blood brings in the osteoclasts, and we're going to start clearing out a space there for what's going to eventually be bone tissue. Okay. So the artery then penetrates the bone collar with the help of osteoclasts and delivers at the same time nutrients and osteoclasts. Okay. Now, you're saying, oh, nutrients. Well, now these cells can survive. No, these cells are already dead. This is the calcified region. This is the region of dead chondrocytes. So they're long gone. They cannot take advantage of this. So all of that has happened already. And now we're bringing in the blood. Now we're bringing in the nutrients, because we need the osteoclasts to remove the calcified dead cartilage. But also we need a lot of building materials. We need a lot of calcium and minerals and nutrients and other things to create the bone that's going to replace this calcified material. That's really a resource-rich material that needs needs a good supply of all of the resources, the energy, the minerals that we need to build bone material. And that can only be there if there is a blood supply within that region. Okay. So the osteoclasts remove the calcified cartilage matrix. And osteoblast begin to produce bone tissue. So the removal of the calcified cartilage, the entry of nutrients provides for the osteoblasts the necessary the necessary resources for them to begin to build the bone tissue. Okay. And once again, this is not a bad series of pictures here. So I'm just going to refer to this. So here is my nutrient artery, and as you can see, it's branching. And so what's happening is we're going to remove the calcified material and replace it with bone tissue. And that process is going to proceed towards the ends of the bone, both ends of the bones at the same time. And so this originally is going to be filled up with spongy bone. And then what we're going to do is we're going to remove the spongy bone. After we build the spongy bone, it gets removed. But the process of removing calcified cartilage is still ongoing, and it's still migrating out to the end. So basically what's happening is we are building, we're replacing the cartilage with bone, spongy bone. At the same time, behind that process, we're removing the spongy bone, therefore creating this large cavity in the center of the bone. And that's going to be the primary ossification center. So none of the textbooks that I have ever seen has gotten that series of pictures right for some reason. I don't know why, but it's unfortunate. Okay, so where are we? So the process spreads. Towards each end of the bone. And this is the so-called primary ossification center. So this is going to form the primary ossification center. All right, so the next step, number 4, is going to be the development of the medullary cavity. Yeah. As ossification moves towards the end of the bones, the ends of the bone, osteoclasts begin to remove some of the new bone tissue. Okay. This will produce a space or cavity in the shaft of the developing bone. And that is going to be our medullary cavity. Okay. Number 5, development of the secondary ossification centers. Development of the secondary ossification centers. So what we have so far is this process. We had that nutrient artery penetrate the bone collar, begin to remove calcified material, replace it with spongy bone. The spongy bone is itself broken down, leaving behind this cavity, and that process continues towards the end. Okay. Now what we're going to see happening is the same process happening here in the ends of the bones, where we get cell death, calcification of the cartilage, penetration of a blood vessel, the removal of the calcified material by osteoclast, the replacement of that calcified material by spongy bone, and that process will proceed outward towards the outer edge. The difference between this process, that is the primary ossification center, and this process, which is being pictured here as the secondary ossification center, is that we will not remove the spongy bone. So there will be no cavity built in the ends of the bones, and that spongy bone will remain there. And then what we're going to see is that finally, towards the very end of this process, we're going to leave a very thin layer of cartilage in place on the outside of the bone, where it's going to articulate with another bone, and a very thin layer of cartilage between the shaft and the end of the bone, and that's going to be our epiphyseal plate. And this epiphyseal plate will provide the ability for this bone to continue to grow in length after we're born and as we grow as humans. This is the material that will be available to add bone growth and length as we move forward in life. So the primary ossification center is going to remove material, create the cavity that you see here. Secondary ossification processes will not remove the spongy bone, leaves it in place, and so the ends of spongy bones are always filled, the ends of long bones are always filled with spongy bone. Okay. So development of the secondary ossification centers. And what we're going to say here is a similar process begins at each end of the bone. Okay. As bone tissue on the outside of the model expands to include the ends of the bone arteries enter and enter the ends of the bones, And then we're going to repeat the process of removing calcified cartilage and replacing it with bone tissue. Except no cavity will be formed at the ends of the bone. So secondary ossification centers do not replace spongy bone. So no cavity is going to be formed. And finally, the last step in this process. Number 6 here, is the formation of articular cartilage. And the epiphyseal plate. So what's going to happen is that the cartilage is replaced by bone tissue, except at the very end of the bones and at the region between the end of the bone and the shaft. So, if I can do this, let's see if I can. So here's the end of the bone, here's the shaft. Okay. And what's going to happen is we're going to form and we're going to have okay, so let's get that. And then in green, okay. So what we're going to have then is our articular cartilage. We're going to have that layer is going to remain in place. And that is going to be our epiphyseal plate. And this is going to be compact bone. And that's going to be spongy bone. So growth is moving out in this direction, and out in this direction, and out in this direction, and out in this direction. At the same time, growth is going this direction. Except that here, we're removing the material as we go forward. There's always going to be a thin line of spongy bone that's being laid down on top of to remove the cartilage. And that growth is going to continue, but behind it, we're removing it so that we end up with this space. And then this area is going to be remodeled into compact bone. And at the end, we leave a very thin layer here at the top of cartilage. The center, or I should say, the center of the end of the bone is going to be spongy, so we're not removing that. But here, we've remodeled everything, so now we have a compact bone as the shaft, the wall, and an open center. So that's my medullary cavity. Okay. And that is how we're going to grow a bone. Okay. And so the final picture that you see in the series from your textbook shows us that completed process, just about anyway. So we're going to leave a very thin layer right here, leave a little thin layer right here. We call that the epiphyseal plate. As I said, we're going to see how that's going to contribute to bone growth and length. And then, of course, this is our articular cartilage, where the ends of the bones are going to articulate with another bone to create a freely movable joint. So that's the process of endochondral bone formation. And the next thing that we want to talk about is bone growth. After we've grown the bone, after we're born, how do those bones grow in length? How do they grow in thickness? And that is going to be actually, as I think about it now, the very last lecture. So will be 4 in this series. The next one will be fairly short because I don't have a lot of material. But I think this is a good time to take a break here. A lot of stuff to consider. And when we come back, we'll consider bone growth in length and thickness. Once you're born and you want to grow taller, how does that all happen? So we'll take a look at that. Thank you very much. I will see you on the other side or something like that. I don't know. We'll see.